Yes, friends. Next question from Dhruva Entertainment and Vidu classes. This question from intermediate second year geology, friends. From unit 4B, that is immune system. What is our question, friends? Explain the mechanism of cell mediated immunity. Friends, cell mediated immunity. You know, friends, what is immunity? It is the ability of an individual to fight against harmful microorganisms is called as immunity. This immunity is of two types, friends. One is cell mediated immunity, another one is humoral immunity. Okay, friends? Now here you have to write about the mechanism of cell mediated immunity. Okay, friends? Now, what is cell mediated immunity, friends? The immunity mediated by the cells is called cell mediated immunity. Friends, here the immunity is mediator. Here who are the mediators, friends? The cells are the mediators. The cells are the mediators for immunity. So the immunity mediated by the cells is called cell mediated immunity. Friends, it is also called as cell mediated response or cellular immunity. Friends, it is also called as cell mediated response or cellular immunity. Friends, very, very important. Here, which cells are responsible for mediation? What are they, friends? T lymphocytes are responsible for cell mediated immunity. Friends, very, very important. T lymphocytes are responsible for cell mediated immunity. Once again, friends, the immunity mediated by cells is called cell mediated immunity. It is also called friends, it is also called cell mediated response or cellular immunity. T lymphocytes are responsible for cell mediated immunity. Yes. The T lymphocytes, like, what are they, friends? Helper T cells, killer cells and macrophages play an important role in cell mediated immunity. Okay, friends, what are the T lymphocytes, friends? The main T lymphocytes involved in cell mediated immunity are helper T cells, friends, helper T cells, killer cells and macrophages. Once again, friends, these are types of T cells, friends. What are they, friends? Helper T cells, killer cells and macrophages. Next, friends, this mediated immunity, cell mediated immunity, is effective against both exogenous and endogenous antigens. Friends, the cell mediated immunity is effective against what, friends? Against exogenous antigens and endogenous antigens. Friends, here I have given the definition of exogenous and endogenous antigens in the yellowish color. Please don't write this in the IPA, friends. Okay? What in by exogenous antigens? These are the antigens enter the body from outside. Friends, what are exogenous antigens? The antigens which enter into an individual's body from the outside are called as exogenous antigens. Example, see, friends, bacteria, virus, fungi, protozoan, etc. They enter into our body from outside, friends. Exo means outside. They are called as what, friends? Exogenous antigens. Come to endogenous antigens. Friends, these are the antigens present in the cytoplasm of human cells. Friends, these are the antigens present in the cytoplasm or inside the body of an individual. They are called as endogenous antigens. What are example, friends? Tumors, viral proteins and proteins of intracellular bacteria. So the cell mediated immunity is effective against what, friends? Exogenous antigens and endogenous antigens. Okay, friends. Now come to our mechanism. Mechanism of cell-mediated immunity. Friends, this cell-mediated immunity has the following parts, steps friends. First you have to know. Number 1, processing of antigen. Number 2, display of antigen. Number 3, recognition of antigen. Number 4, production of activated T cells and memory cells. Number 5, role of activated T cells. Number 6, role of CTLS. Friends, you have to remember these 6 steps, friends. It is easy to us to write in the exam. What are those 6 steps? Processing of antigen, display of antigen, recognition of antigen, production of activated T cells and memory T cells, role of activated T cells, and role of CTLS. Okay, friends. Now, first we will see the processing of antigen. See, friends, important friends. Processing of antigens. It is an immunological process that prepares antigens for presentation to T lymphocytes. Friends, when our body has antigens, these antigens has to be processed, friends. It is an immunological process. In this process, antigens are prepared for what? For presentation to T lymphocytes. That is called as what, friends? Antigen processing. See, friends, T cells, T lymphocytes can't recognize the free circulating antigens. They won't recognize without processing. So, when the antigens are processed, then only the T lymphocytes recognize them. So, T cells cannot recognize the free circulating antigens. These T cells can recognize antigens when antigens are processed and presented by APCS and ACSS. Friends, APCS and ACS. Friends, what is APCS and what is ACS? Exogenous antigens were processed by APCS. Friends, APCS means antigen presenting cells. Friends, you know what are exogenous antigens? These exogenous antigens were processed by whom? By the APCS, that is antigen presenting cells. Example, friends, dendritic cells and macrophages. Friends, these all are important for MSET and NEET. Dendritic cells and macrophages are called as APCS. APCS processed exogenous antigens. Okay, friends. Then only the T cells recognize them. Next, friends. Endogenous antigens were processed by ASCS. ASCS. What is ASCS, friends? Altered self cells. Example, friends. You forget about this, friends. Dendritic cells and macrophages. Okay, friends. That is about the processing of antigens. Come to display of antigens. After processing, these antigens has to be displayed, friends. After processing, these antigens were displayed by APCS and ASCS on their cell membrane. What is the first step, friends? Processing of antigen. Second step, they have to be displayed. Displayed by whom, friends? By whom? APCS, that is antigen presenting cells, and antigen, friends, ASCS. Okay, friends, altered self cells on their cell membrane. Next, friends, recognition of antigens. Recognition of antigens. Processed and displayed antigens are recognized by T cells. See, friends, without processing, T cells won't recognize them. So, the processed and displayed antigens are recognized by whom, friends? By T cells. Very, very important, friends. Now, after recognition, what is the role of the T cells? Production of activated T cells and memory T cells. Binding of T cells to the APCS and ASCS causes production of activated T cells and memory cells. After recognition, these T cells, what happened, friends? They produce what? Activated T cells and memory T cells. Activated T cells and memory T cells. Next friends, what is the role of activated T cells? These activated T cells secrete interleukins, friends. These activated T cells secrete what, friends? Interleukins. These interleukins transform activated T cells into effector cytotoxic T lymphocytes or killer cells. Friends, what is the role of activated T cells? These activated T cells secrete interleukins. First point, friends. These interleukins transform activated T cells into what, friends? Effector cytotoxic T lymphocytes. TLCS, friends. CTLCS. Cytotoxic T lymphocytes. Or you may call it as killer cells. Next, what is the role of CTLS, friends? What is the role of cytotoxic T lymphocytes? These cells attach to the infected cells and release perforins and granzymes. Friends, we have a 2 marks question. Perforins and granzymes. Perforins make pores on the infected cell. Granzymes enter into that cell and they destruct it, friends. So, these cells means what, friends? Cytotoxic T lymphocytes attach to the infected cells and release perforins and granzymes. What are perforins and granzymes, friends? They are enzymes. These two are enzymes. See, friends, perforins form pores in the cell membrane of infected cell. You know, friends, you know the meaning of the perforins, role of the perforins. At the same time, then granzymes enter infected cell through the pores formed by the perforins. These granzymes secrete proteins like, friends, capsages. Here you have to remember capsages and destruct the infected cell. That is called as apoptosis. Already we read about these two perforin functions of the perforin and enzymes in a 2 marks question, friends. What is the difference between perforins and granzymes? Once you go through it, friends. Okay, friends. Now see here an image, friends. For NEET and MCET, friends, what is apoptosis? It is a process of programmed cell death. See, friends, CTLs means cytotoxic T lymphocytes or killer cells. IFNs, interferons. These are antiviral proteins produced by virus infected cells and involved in protecting the neighboring healthy cells from the viruses of infected cells. Friends, here it is not, it is the, friends. What are IFNs? Interferons. Interferons are antiviral proteins produced by virus infected cells and involved in protecting the neighboring healthy cells from the virus infected cells. Very, very important, friends. Next, what are IFNR? Interferon receptors. What are TCR? T cell receptors. What is MHC? Major histocompatibility complex. What are interleukins? These are one type of cytokines secreted by TH cells. They bind to cell surface receptors and stimulate phagocytosis and cytolysis of the infected cells. Okay, friends? This is about what, friends? Cell-mediated immunity. Once again, you see the mechanism of cell-mediated immunity, friends. Once again, friends, it has the following process, following steps. What are? Number 1, processing of antigen. Number 2, display of antigen. Number 3, recognition of antigens. Number 4, production of activated T cells and memory cells. Number 5, role of activated T cells. Number 6, role of cytotoxic T leukocytes. Okay, friends? This is about what, friends? Cell-mediated immunity. Cell-mediated immunity. Okay, friends? Thank you. Good day.